helped a lot if we have some understanding of suborganismal mechanisms. The second take-home message is that within the organism, feedbacks really matter. And the third take-home message is at the ecological level, feedbacks really matter. So in even summarizing the nutshell, suborganismal processes sometimes matter. Feedbacks both within the organism and at the ecological level always matter. So that's what I'm now going to take a little bit of time to elaborate on. And I'm going to start by reminding you of a slide that Valerie put up yesterday where she talked about the scope of existing models. And by a nice coincidence, the section she circled as being the focus of the CREAM project, in which she was one of the leading participants, is in fact the area that's largely covered by what Volker and I are going to talk about this afternoon. And oops, I pressed the wrong button. I'm going to focus primarily on the use of dynamic energy budget models, and by the end of the lecture I'll have changed the nature of this arrow just a little bit, and Volker is going to talk about IBMs, but I'm sure again he'll make connections to other bits as well. And well, I think I'll leave the remaining comments till a little bit later. And I have a slide which is a variant on the slide that it seems just about everybody here has put up, that defines in many ways the challenge that we're trying to address as modelers, but I've got a slightly different spin on it. Like many previous speakers, I've recognized that we're in a situation where we have an increased access to information from high throughput screening, high content screening, and whatever else you care to call it. Lots and lots of short-term data, primarily at the level of cells or at very small organisms like bacteria and yeasts. A few, for instance, on zebrafish embryos and so on. And I read an amazing paper a little while back where actually the full life cycle of a copepod was studied in a 96 well plate, where some tiny copepod that only grew to about 0.3 of a millimeter. But still, oops, I keep hitting the wrong one. But still, there is huge amounts of data, a increasing ability to get it cheaply at this level. We move up to whole animal testing and ecological investigations. It's expensive, there's ethical challenges, and there's practical challenges. And the challenge for theorists that I'm going to talk about is to extract maximum benefit from these cheaper or more rapid studies in order to prioritize, guide, design, and interpret ecological studies and inform ecological risk assessment. And the word predictive is not there, because I have no confidence of our ability to use this type of information to actually make predictions at this level, but I have a lot of confidence that we can advance methods that will use the low-level information to help us understand and focus our studies at the higher level. And I hope it's beyond the type of triage that is represented by the sort of tier 1 testing. The role of models to try and make that connection perhaps a little more mechanism driven. I want to make some remarks about how we as modelers attack population dynamics. And with change in terminology, what I'm saying here applies to both individual-based population, what are called individual-based models, and what are often called structured population models. But I would emphasize that they're actually both individual-based models. I'll stick my neck out and say that all good population models are individual-based. And what I mean by that is that the parameters in a population model, even if you set it up as a population, should make sense as some kind of aggregate of the properties of individual organisms. Straight away you run into difficulty with that. With that definition, the logistic equation that appears in every undergraduate ecology class has one parameter, the intrinsic growth rate, which is okay, and one parameter, the carrying capacity, which is a horror. And I leave that thought with you. But certainly everything I'm going to talk about, and I think that Volker's going to talk about, is based on the perspective that a population is a collection of individuals following certain rules. In the work that I'm going to talk about, which involves these so-called structured models and simplifications of them, it's useful to identify three types of variable. Organisms live in some environment, so you need a set of variables that characterize the environment. I'm calling these E-state variables. An individual organism has to be represented by a set of variables, and much of what this lecture is about will be about choosing these variables and trying to end up with half a dozen rather than several thousand. And then for one approach that is used, the so-called structured models and the models that derive from them, we also need a way of characterizing the population. And this is done using variables which are p-state, which are called p-state variables. They represent some, well, they are some representation of the makeup of the population, how the I-state variables are distributed among individuals. It might be population size, age structure, some broader distribution. But the important message here is that moving from individual to population, whether you do it in the context of analytical models like the structured population models or individual-based models, it's essentially bookkeeping. It's just bookkeeping which can be relatively simple in some situations and very complicated in others. And for that reason, for me, the art of formulating population models is about understanding the individual organism, understanding the environment, and about understanding the feedbacks. And for that reason, I've written this little triad at the bottom of the slide here, which appears in various population dynamics books, but I've put a big red emphasis around the individuals. And that's because if we get the individuals right and their interaction with the environment, and we know the feedbacks, then the population dynamics is simply a matter of feedback, and for a matter of bookkeeping. And for that reason, I'm going to say nothing about the technicalities of putting together population models in this talk. I've said in words what is in this slide, that there really are two approaches to modeling population dynamics. And I've written down the traditionally used names here. For me, they're a misnomer because it's really two types of individual-based models. There are what are often called individual-based models, like agent-based models that are put in a computer. And there are these models that use a more mathematically formulated, a more formal mathematical structure. In many ways, they represent a mean field characterization of the dynamics that is described here. If you want more on structured population models, there's a huge collection of good papers by Hans Metz, Odo Diekmann, and André de Roos, and some very useful material on André de Roos's webpage. And I'll later refer to a superb book by de Roos and Leonard Persson that came out about 2 years ago that has a significant relevance for what we're doing here. I'm now going to immediately narrow the scope of what I'm talking about, and I'm going to talk about one particular way of describing what I call the I-state dynamics, one description of individual organisms, and that's the one that has been mentioned several times already, the dynamic energy budget approach, much of it developed by Bas Kooijman. The definition of a dynamic energy budget model is it's a model that describes the assimilation and utilization of energy and elemental matter by living organisms. In the formalism that I'm going to describe today, the growth, reproduction, and mortality of an organism are related to its food intake by a set of differential equations. Then the population dynamics are just worked out applying the recipes. And I'm going to focus on one particular model, though most of what I say is not restricted to this model. It's just the one we've used for a lot of work. It's a so-called standard DEB model developed by Bas Kooijman. It describes all life stages of an animal. It assumes that there are 6 I-state variables and around a dozen parameters, about which I'll say more later. And I've listed some references here. If anyone can bear to hear my voice for longer than the 45 minutes we're talking about, there's a set of lectures that introduce DEB theory on the CEIN website. That's the Center for Environmental Implications of Nanotechnology. And if you don't want to write that down fast, but I'll give anyone the link. Or if you just Google CEIN and NISBet, you'll get there. Before we go any further, a warning about the jargon that people working in DEB theory use. And it's a sufficiently important problem that I want to highlight it, because many people think DEB theory is nonsense because of somewhat idiosyncratic use of certain words. I put up a quote there from a famous 19th century mathematician who also was the author of Alice in Wonderland and Through the Looking Glass. Some of you may be familiar with it. Humpty Dumpty, when I use a word, it means just what I choose it to mean, neither more nor less. And it ends, the question is which is to be the master, that's all. And for the next 5 or 10 minutes, I'm the master in this. I say what words mean, but in the bigger picture, there is a change, and it's Bas Kooijman chooses. And you'll see in the next slide why I put this warning up. Because here is a schematic of the simplest dynamic energy budget model of an individual organism. It represents a concept where food from the environment is absorbed, some fraction is excreted, some fraction immediately enters a component called reserve, and then the energy from the reserve is mobilized and used for either growth, meeting maintenance, development, which involves increasing maturity, or for an adult for reproduction. In this framework, structure is nothing to do with bones and skeleton and shell or whatever. Structure is defined as biomass that requires maintenance, and reserve is defined as all biomass that does not describe maintenance. So all we're saying here is that for the purposes of this talk, we're not allowed to challenge it for the next few minutes. We use this terminology: forget what you thought reserves were, forget what you thought structure was. The structure requires maintenance, reserves don't. Mathematically, what really distinguishes Bas's model from many other models that characterize the flow of energy in living organisms is recognition of the importance of homeostasis. That's feedbacks within the organism. And in particular, the rate of mobilization of energy is determined by the requirement that the reserves be regulated around some fixed fraction. And that's what contains, that's where much of the elegance and power of the model derives from. That representation characterizes growth and reproduction. There is also a characterization of mortality, and here I mean the physiologically related components of mortality. And I took this somewhat busy picture from a set of comments on Bas Kooijman's website on his book. And I should mention that that is also a superb resource if you're interested in DEB theory and you look at Kooijman's book and you find it incomprehensible. There's a huge file of comments where he's taken a huge amount of effort to respond to points that people have raised. And frequently they're easier to read and much, much sharper than the book itself. I want to illustrate how the model describes mortality by one example. Many stressors have their effect on cells through oxidative damage. So some process leads to the production of reactive oxygen species, which in turn have impacts. The idea in DEB is that we have some process which we can which I'm representing here just by an example as ROS, reactive oxygen species. These in turn produce damage, but there is some feedback from the damage to the ROS. In the example in Kuyman's book, this is positive feedback, but in many cases it will be negative feedback, and there is a whole area of research to be explored, which we are ambitious to do if we can get some resources, that involves exploring these feedbacks further. But that leads to the introduction of two further variables, a variable here which are commonly called damage-inducing compounds, and a variable here that is called damage. So what that means is that the model now gives us a representation of an individual animal with 6 state variables. The first 4 of these came from the description of growth and reproduction. The next 2 came from the description of mortality. The precise number of parameters depends essentially what you're prepared to regard as a fact and what you're prepared to regard as a parameter. But that's why I said of the order of 12 parameters. And if anyone wants to talk about parameters later, I would love to do so. The model then takes the form of a set of ordinary differential equations. They're just balance equations, but the expressions for the fluxes have to take account of these various homeostases. I've written the equations for 4 of the variables here, 4 of the fluxes. There's 2 extra equations for ordinary differential, for the damage-inducing compounds and the damage. But for this talk, all that matters is that we're dealing with a set of messy ordinary differential equations, and there's nothing particularly interesting or challenging about them. So what has any of this got to do with ecotoxicology? The first important feature, which again I can elaborate on, is that the model is built on very general processes that apply across everything from smallest animal to the largest animal, and although I won't talk about it, can be extended to plants and simplifies when we're dealing with microbes. And there are rules within the model for transferring parameter values across taxa, which means that there is the potential for extrapolating from model organisms to other organisms. At the cost of extra I-state variables, it is possible and indeed very valuable to incorporate toxicokinetics into the model, so the models can describe bioaccumulation. It's possible to include toxicodynamics, and indeed this is the key to applying to current methods for applying the model to ecotoxicology. The idea is that the external or internal concentration of a contaminant impacts the model parameters, not the end process. So we're not talking about going all the way from suborganismal processes to things like growth and reproduction. We're talking about going all the way from suborganismal processes to depth parameters, and then the model takes us the final stages. If you want to get a readable introduction to depth theory focused on ecotoxicology, there is this wonderful ebook written by Chanling Yager, which is a free download, and several people here have commented how useful they've found it. I think it's a really terrific piece of writing. So I now want to say just a little bit about population dynamics derived from this framework. And to start, I have to jump back to some of the material that I use when I'm teaching my upper division ecology class, which is how traditionally we describe changes in a population. Suppose we have a population where there is some fraction of individuals that survive to age A, some fraction that of offspring that are produced by an individual age A. Then there is a famous number called R0 that you can calculate, which is the average number of offspring produced by an individual in its lifetime. In the textbooks, it's always called often called the net reproductive rate, even though frustratingly it's not a rate, it's a number. But in continuous time, this just generalizes to an integral. And the nice thing here is that our DEB model gives us in a constant environment, this quantity beta, this quantity S, so we can compute the lifetime reproductive output for a model from the Deb model, and if there is a stressor, we can compute changes in that quantity. And I'll illustrate this process in action with reference to some work that Eric Muller, who's sitting at the back, led working with PhD student Shannon Hanna at UCSB. Shannon did a bunch of experiments on the response of marine mussels on exposure to zinc oxide nanoparticles, and did a mixture of short-term and more extended studies on the quantities that are listed here. We used these to estimate the parameters, or some of the parameters, in the Deb model. We couldn't get at all the parameters in the model, but here is where one of the nice features of the model came in. There's been a lot of work on mussels done by other people, and there is a web resource called Add My Pet, and you just need to remember the name Add My Pet and Kuyman and Google it, and you will get to this resource with estimates and measures of confidence of Deb parameters for around 302 species that are available. So you can supplement the information that you get from your experiments with information from other work and at least get a first cut at the parameters. And then, of course, there's room for another whole lecture on ways of using that information to refine your estimates, but I don't want to go there. Doing that, the result was the following. This is a graph where the x-axis is food availability. The y-axis is an inverted measure of the nanoparticle concentration that was supplied at the start of or supplied during the study, and the curve is an approximation to an EC50, the level at which the net reproductive output was dropped by 50%. And the take-home message was that it was possible to estimate EC50s for the individual physiological rates, like a feeding rate, and then it was possible to estimate it for the population growth rate, and the EC50 was much, much smaller for the population growth rate for the individual growth rates. And that's because an organism in that situation has a double whammy. Feeding more slowly means that it grows to a smaller maximum size. It also means that because it's feeding more slowly, even size for size, it produces fewer offspring. So there is a potential for amplification of the effect, which you can quantify very nicely with this type of model. But only when we're dealing with a few situations, in particular, for instance, involving a threatened species or endangered species, are we really interested in just the long-run population growth rate? Because lesson 2 in an undergraduate ecology class tells you that populations are regulated, they don't grow exponentially forever. And so feedback from the organisms to their environment really matters. And for this, there are a bunch of mathematical techniques available if we make model simplifications. You can, of course, take the structured model straight away and work with it, or you can set up an individual-based model and work with it. But it's really useful to know about these simplifications. One is that if you assume that all the physiological rates in the Kuyman model scale with some measure of biomass, perhaps the structural biomass in the DEB model, but it's flexible what you do. And more heroically, you assume that all organisms experience the same risk of mortality, an assumption which it's remarkable you can quite often get away with, even though it seems absurd. Then you add in some ordinary differential equations describing the environment. Then your complete dynamical system is described by a set of ordinary differential equations. And in some cases, you come up with a model that looks very much like one put together by Yotzes and Innes, perhaps 20 plus years ago. And there is this superb book by de Rosen Persson, which follows that approach and structured models of increasing sophistications in interacting populations through, and there is a set of web lectures by Andre where you can again get an introduction to that material. If you really don't like the idea that mortality rates are the same across stages and it eventually reaches a point of absurdity, a larval fish compared with an adult, you can break the model up into stages, in which case you get sets of delay differential equations, which are just like ordinary differential equations. Instead of evaluating some of the variables on the right-hand side at the current time, you do it at a previous time. You need some caution when implementing them numerically, but there's plenty of good software available. There is a reference here to a book chapter of mine that summarizes this approach to population dynamics. And again, I'm willing to bore anyone to tears who wants to talk about them, for I spent an embarrassingly large proportion of my research career working with these delay differential equations. And that third reference is a book that makes use of them within what I would call mainstream ecological theory. I want to illustrate the use of this simplification in a situation for which it's probably quite readily applicable, and that's to a study of microorganisms. And this is another nano example. It involves a study of the effect of one particular type of nanoparticle, cadmium selenium quantum dots, on one particular strain of a bacteria. These particles are used in electronics. And the question of interest was, is there a difference between organisms experiencing a dose of cadmium from the exposure to the quantum dots versus what would happen if they simply experienced a dissolved cadmium salt? And in other words, does the fact that it's nano matter at all, or does everything we already know on cadmium toxicity carry over? And to do this, the people we collaborated with, whose names appear in the water at the bottom, John Priester, Trish Holden, were the experimentalists, did a study where equal amounts of cadmium were provided in these two forms to a large number of bacterial cultures. And we constructed the modeling using a somewhat elaborate scheme that I've described there. And rather than describe it in detail, I'm going to do it in steps. And the first was, we only modeled the effects of exposure to cadmium. So this was like the kind of bacterial modeling that could have been done absent any information 20 or 30 years ago. Interestingly, we discovered that even the control curve, that's the black curve there, where there was no stressor added, didn't fit the traditional microbial ecology models, and we actually had to introduce a new feedback which probably represents stress from living in a 96 well plate rather than in a chemostat. The end result was we worked with a variant of Bass's model. We took Koymann's model. We were dealing with bacteria, so we didn't have to worry about reproduction and quantity related to reproduction. We kept the damage-inducing compounds in the damage. We had dreadful problems describing the startup of these cultures, and anyone who's worked with microbes knows that there are problems with lag phase and all the things that cause lag phase. So we introduced a new variable to describe the lag phase, and then we had no idea what was causing the damage in the 96 well plate. So we went non-mechanistic and invented a variable called environmental degradation, which sounds very grand, but it just means extra variable to cause the model to fit the data. And but it's not quite as bad as that, but it's not much better. The end result was we finished with a model that had 6 parameters plus a variable number of other parameters depending on the precise toxicodynamic model that we used. We then were able to use the model in a situation where we assumed different toxicodynamics. We assumed the toxic effect operated in this parameter, this combination of parameters, and so on. and then used statistical fitting methods for model selection, and were able to conclude, in this case, which model best fitted the data. It happened to be a model where there were simultaneous impacts on assimilation rate and mortality. That was substrate assimilation rate, but the details don't matter for this. The point was we were able to perform the model selection and get at some mechanisms. And very optimistically, we thought we would like to do the same thing now with the quantum dots. So we just went through and we imposed one rule. We said there's a whole range of possible things that people who know about nanomaterials tell us could potentially happen. And all you really need to know about this, see about this picture is that it's complex and there's a lot of potential mechanisms going on. We then thought, okay, we'll put all these in the model and we'll follow exactly the same procedure as before, fit the data, but with one rule of the game, that the effects of cadmium toxicity in the environment were not allowed to change. So the particles are dissolving at known rate, the cadmium ions are appearing in the environment. We're not allowed to change the model parameters that do that. We want to fit the new processes. We thought that would be fine. Actually, what happened was a load of our models were equally good. We'd hoped to separate the models into the good, the bad, and the ugly. We got rid of the ugly, but we were left with the others. So we actually needed more information. And this is why I've included this slide. We focused on an idea from basic Deb theory, which was that these somewhat abstract damage-inducing compounds could be related directly to measurements of ROS. And it seems everyone in the nano world that is looking at effects of nanoparticles on cells gets at least some assay that is representative of something related to ROS, and these come with different levels of sophistication. So we actually had used the traditional information. We hadn't used the ROS information. So now we invented for ourselves a metric which was the ratio of ROS production to in the quantum dot exposure to the ROS production in the cadmium studies, and followed that metric over time. And that actually proved to have powerful discriminating capability. We were actually able to separate one model from the other. So the take-home message was, and this is, I realize, a messy example to make it, by adding just a little bit of molecular information, we were actually able to tease out some factors that were impacting population dynamics. And that's a very positive message. The caution that comes with it is that the validity or otherwise of this conclusion depends on us getting the feedbacks correct between the ROS and the damage, in particular the extent to which a ROS is internally regulated in the cells. In other words, we need to know a bit more physiology to evaluate our own work, and like I say, that's a direction we want to go. So that's the positive part, and the cautionary part is that it's based on a portion of the model that needs a further exploration. I want to end the presentation by showing you one piece of work that actually connects into what Volker is going to talk about relating to interactive populations, because I've demonstrated the importance, I think, of physiological feedbacks, homeostasis within the organism, but ecologists for a century, or 3/4 of a century, at least perhaps more, have been working with models where that involve what I'll call ecological feedbacks, where there are nonlinearities characterizing interactions among species. And I want to show one particular example where that matters, which involves my favorite organism, Daphnia, and its experiment. It's worked by Ben Martin, who is over at the left-hand end of that road, who you've heard a couple of times giving a report from one of the breakout groups. And this was an attempt to construct a population model for Daphnia interacting with their food resource based on experiments by Thomas Preuss as part of Ben was part of the CREAM project, based on experiments by Thomas Price on the effects of one particular contaminant, which appeared to these slides, it's come up very slowly, based on a set of experiments on individual organisms, which essentially showed that that particular stress had no effect on growth and reduced reproduction. And a model was built in that, and there is a paper that I've given the reference to there. But most stressors don't produce a simple effect. It's much, much commoner to see an effect on growth and reproduction, and to get different types of information on the relative magnitude of these effects. So what Ben did was to then go and say, instead of that representation, let's look in a model at all the places where the stress could act. It could act here, it could act on the components of maintenance, it could act on growth, it could act on reproduction. And then looked at studies, and then used the model of individuals to calculate the level of contaminant that would cause a reduction of a specified number of percent in the long run growth rate. That's the quantity that you would get without feedback. And then the plot on the right shows the percentage change in biomass due to exposure compared with the percentage change in abundance due to exposure. And these are interacting populations. And the take-home message is that if the primary impact was here, so if you like, if the pathway impacted was at this point in our simplified scheme, then what you would see would be a reduction in biomass, but if anything, a marginal increase in numbers. So the population consists of a slightly increased number of smaller Daphnia. If the impact is here, as in the Price experiments, then you see very little change in biomass, but a substantial reduction in abundance. If it's in two other places, you get a pretty much proportional increase in each. In other words, figuring out where in the life cycle the impact occurs has huge ecological consequences. And I could translate that, and if I'd made the slide up at lunchtime rather than last night, I would have. If I translated that into this morning's talk, it effectively says AOPs matter, because it's telling you which, with a slight reinterpretation of the word, it's telling you that which pathway it is matters, really has a big effect on the ecological consequences. Ben did something else that was very nice. He took took one of the classic textbook population ecology models, the Yodzas-Innes model that I've referred to earlier. Remember, it has an individual-based underpinning, so it's a nice model in that regard, and asked what would it say would happen in this case, and it was simple direct proportion like this. So what that says is that the classic picture is capturing some of the story, but if you want to get further, you actually have to penetrate to what individuals are doing, and that involves getting under the skin of the organism. So to pull it all together, I want to again return to Valerie's opening slide. Valerie presented us with this glowing picture of the future at the end of her lecture. And oops, that's what I was trying to get. And I think what I've tried to do is take the picture that she described in the top part here and say that actually the Deb approach is actually giving us some broken reach down here. It's a bit fragmentary, and although I haven't talked about it, we actually have some work that demonstrates you can make some connection to the ecosystem, and that's because one of the features that I didn't discuss in the Deb model is the fact that they have excretion products, and these in turn impact ecosystem processes. And this means that I'm actually reasonably confident that without completing the straight line and the predictive part, we can make significant advance on our mechanistic understanding of the complex systems, and that we're moving forward in making these connections. And that's where I'll stop. Thank you.